Sheep are dying with clinical signs including edema of lips, nose, face, submandibular area, eyelids, and sometimes ears, congestion of mouth, nose, nasal cavities, conjunctiva, and coronary bands, and lameness and depression.
Samples that you would collect to send to the lab would be blood samples from live animals with clinical signs. In dead animals, samples of the liver, spleen, red bone marrow, or heart blood can be taken. In aborted newborn sheep, samples of the blood and, if possible, of the lung, spleen, brain, and serum should be collected. Samples Transporting and preserving the specimens should be done safely and responsibly by following basic packaging requirements. The sample should be put in a primary receptacle and then in a secondary packaging like a sealed plastic bag. The secondary packaging should then be put in a tertiary outermost layer like a cooler bag.
Now, there are four approaches to confirming a viral infection in the laboratory. The first one is antibody assays. An acute or recent infection may be confirmed by demonstrating the presence of specific immunoglobulin M in a single serum sample or showing a rise in specific immunoglobulin G. In general, the presence of immunoglobulin G and the absence of immunoglobulin M is indicative of past infection or immunity. Now, there are four approaches. The second approach is the use of electron microscopy. Historically, the electron microscope was very useful in defining the morphology of many human viruses. However, it's not a tool that is routinely used to identify viruses in a diagnostic setting because viruses are usually present in very small numbers in clinical specimens and other contaminating material tends to obscure their presence. Viruses can only replicate in living cells. Therefore, to culture them in vitro, one must provide them with living cells. Replicating viruses change the appearance of cells to induce a cytopathic effect. Different viruses cause different types of cytopathic effects, allowing one to identify the virus using the type of cytopathic effect. Another way to identify a virus growing in a cell culture is to add fluorescein-labeled monoclonal antibodies to likely viruses to the cell sheet and examine under a fluorescent microscope. Nucleic acid amplification techniques such as polymerase chain reaction can be used to detect viral genomes in clinical material. To detect RNA, an initial reverse transcription step is performed, converting RNA to cDNA, after which PCR can be performed. Molecular assays are very sensitive, thus they can also be used to measure the amount of virus in a patient sample. Viruses are obligate intracellular parasites, so they depend on a host for their survival. They cannot be grown in non-living culture media or on agar plates alone. They must require living cells to support their replication. Cell culture is mostly used for identification and cultivation of viruses. Cell culture is the process by which cells are grown under controlled conditions and is mostly used for identification and cultivation of viruses. Cells are grown in vitro on glass or treated plastic surface in suitable growth medium. On incubation, the cells divide and spread out on the glass surface to form a confluent monolayer. Viruses can also be cultivated using animal inoculation and inoculation into an embryonated egg.
Polymerase chain reaction is the best method for growth of blue tongue virus. It is carried out in an instrument called the thermocycler. Firstly, the DNA of the virus is denatured by increasing its temperature so the DNA can separate. Once separated, the DNA is cooled, primers are added and allowed to bind to the single DNA strands. After attachment of primers on each DNA strand, a DNA enzyme, Taq polymerase, copies the DNA sequence, forming two double-stranded sections of DNA. Taq polymerase is helpful for laboratory tests of this nature since it does not break down at high temperatures required for PCR. The process is repeated again. Heat is applied to the two double strands of DNA to separate them into 4 single strands and cooled again for addition of primers and Taq polymerase. The 4 strands become 8 in the next cycle and the 8 becomes 16 and so on. Within 30 to 40 cycles, billions of copies of original virus DNA can be produced and used for molecular diagnostic tests.
The morphology of a virus is usually determined by the use of an electron microscope by using a stain. There are 3 types of stain:
shadow casting, negative stain, and positive stain. Once you have viewed the sample through the microscope, you can compare its morphology. There are 4 basic morphology types: icosahedral, helical, complex, and binary. The blue tongue virus has an icosahedral morphology, consisting out of an icosahedral capsid enclosing double-stranded RNA genome.
The eclipse phase is the 4th phase in virus replication. It's the phase during which the virus has entered the cell but is not yet infectious. During this phase, the virus gains control of the host's synthetic machinery and produces components for viral assembly. Intermediate host of the blue tongue virus, the viral titer decreases in the eclipse phase due to the inactivation of some virus particles in the hostile environment of the gut and excretion before viral proliferation in infected gut cells has taken place.
Mutations are common in blue tongue virus due to the RNA polymerase lacking an effective proofreading mechanism during the replication process of the viral genome. This ultimately results in the generation of quasi-species or serotypes during the infection process. The most common type of mutation to occur in the blue tongue virus are point mutations, specifically substitutions, where one nucleotide is replaced by another. Mutations are acquired both randomly and biasedly during the transmission phase of the virus, when the virus moves from its insect host to its ruminant host. These mutations show a temporal increase along its course of infection. The virus does, however, contain consensus sequences which remain stable throughout the transmission cycle. Mutations occurring in blue tongue virus have ultimately resulted in 24 distinct serotypes being formed. This results in an increase in the variability of surface antigens present, thereby affecting the virus's ability to react to specific antibodies of certain host species. Nucleotide substitutions within the blue tongue virus are considered to be an important evolutionary mechanism, as it is the main source of generating quasi-species through genetic drift events. Quasi-species allow for significant adaptive potentials of the virus through the selection of mutants displaying the highest fitness. This ultimately allows for rapid evolution, increasing the virus's ability to evade host defenses. The viral structure protein VP2, responsible for viral attachment and entry into the host cell, as well as non-structural proteins NS3, NS3A, controlling replication, undergo frequent A to G substitutions during transmission. These substitutions have been found to introduce premature stop codons within the protein sequences, therefore rendering it non-functional, leaving the virus unable to attach and enter its host.
Vector-to-host transmission takes place via the saliva of biting midges from the culicoid species. Biting midges are the carriers of the virus, and they feed on the blood of the ruminants that they bite. The virus has a high affinity of blood cells and will quickly spread into the blood of the ruminants and infect the host. The virus is not contagious and cannot be spread through contact between animals. There must always be a vector for the virus to spread. Host-to-vector transmission takes place when uninfected biting midgets feed on the blood of infected ruminants and get infected. The virus is shed into the environment when infected biting midgets disperse from infected areas to uninfected areas in the environment. The biting midgets in their new and uninfected environment bite uninfected animals and pass the virus on to these animals. Vectors. Antigenic disguises. Antigens disguise themselves in the cells of the body. To the immune system, the pathogen appears as normal cell material and the immune system does not recognize it. Thus, the intracellular pathogen escapes the host immune response and stays healthy. Immunosuppression. When pathogens suppress the immune response towards themselves as well as towards unrelated pathogens, it is often observed when pathogens hide somewhere else than in the blood for a relatively long period of time. It will first grow and wait for an opportunity when the immune system is compromised before it starts to multiply and cause an infection in the host. Specific immune response. Pathogens hide in certain areas of the body where cells are not protected by the immune response. Antigenic variation. Each time that the pathogen infects the body, it will vary its genes to form different proteins that are not recognized by the host's immune response. The pathogens will neither be recognized nor attacked by the antibodies that was formed during the previous infection of that specific pathogen.